Okay, thank you very much. I appreciate having the chance to be here today. I don't remember if I was given a title or not, and based on the title of the session of major players, I decided to assume that you were not talking about me, and you were actually talking about things that were involved in valve disease. So I decided to talk about my approach, which has been to develop in vitro models for investigating heart valve mechanobiology and pathology. So the things that I think about in terms of what is important to consider, well, one of them, and really the driving factor, is the valve microstructure. So due to the layered microstructure that you are all familiar with, whether it's the aortic valve or the mitral valve, having layers of collagen on the inner curvature layer and elastin on the outer curvature layer and glycosaminoglycans in between, these give rise to the heterogeneous mechanical behavior of the valve, and also provide the valve cells with heterogeneous microenvironments, whether it's the valvular endothelial interstitial cells or the valvular endothelial cells. And so I also think about how the structure and the mechanics of the valves are altered in disease or in heart defects. So clearly the mechanics of the valve are very altered, and overall structure very altered, whether it's a myxomatous mitral valve disease or calcific aortic valve disease, or in congenital heart defects. So the lens through which I look at all this is through biomechanics and mechanobiology. And so some of the things that I think are really important are the anisotropic characteristics of the valve. So with the circumferential collagen fibers really dictating that mechanical feature, as well as the nonlinear mechanical behavior with a high extensibility initially, leading to high stiffness later. And a lot of that is due to the mechanical characteristics of the extracellular matrix, but the interstitial cells are also very mechanically active. They contract, although somewhat slowly. They express smooth muscle alpha actin if they are activated, and the activated VICs have a lot of extracellular matrix synthesis. And then, again, going back to the heterogeneous layered structure, not only does this give us the desired mechanical behavior, but also provides the cells within with a range of pericellular stiffnesses due to different extracellular matrix and different biochemical ligands on the extracellular matrix that the cells can bind to. So the driving questions for me are, can we develop model systems to investigate the biomechanics and mechanobiology of the native valve? And a lot of this has to do with just looking at relationships between extracellular matrix to structure and the overall mechanical behavior, as well as looking at the effects of these conditions on valve cell behavior and how this is different in valve disease. And so we have been using biomaterial and tissue engineering platforms like bioreactors. And then another aspect is, if after developing these biomaterial platforms for investigating mechanobiology, can aspects of the model platforms be translated to improve tissue engineered valves? So I'm going to mostly cover more recent work. So I'm going to start out with our investigations of valvular endothelial cells, hemostasis, and calcific aortic valve disease. And so endothelial hemostasis, just overall, not just in valves, is a balance between antithrombotic and thrombotic proteins that are expressed by the endothelial cells and control clotting. And so there's a number of antithrombotic components like the von Willebrand factor cleavage enzyme, ADAMTS13, balancing von Willebrand factor and making sure that it doesn't get too large. And other things balance each other, like tissue factor plasminogen inhibitor and tissue factor, tissue plasminogen activator, and PI1 are a lot of the things that we've been looking at the way valvular endothelial cells behave. So this is a fundamental characteristic of endothelial cells, but one that is not really studied very much in valves. So we've been looking at how valvular endothelial cells taken from different ages of animals produce these thrombotic and antithrombotic proteins. And just to summarize a lot of busy data here, just as an example, just looking at, for example, tissue factor plasminogen inhibitor and tissue factor, you can see that there's a reduction in the gene expression of TFPI with age. There's a hint of an increase in tissue factor with age. We see similar sort of balances between a reduction in age of some antithrombotic factors and an increase with age of more thrombotic factors. So just overall, there seems to be a shift towards more thrombotic proteins accumulating in the valves with age. And this doesn't mean that our valves naturally thrombose with age, but in the condition of maybe an injury, this could certainly cause a cascade of events. And in particular, we do find that the gene expression of von Willebrand factor does go down with age, but if you immunostain the tissue for von Willebrand factor, you can see that it actually does accumulate within the tissue. So even though the cells aren't making it anymore, it's still there. So we then took cells that had been valve endothelial cells that had been stimulated with histamine to make ultra-large von Willebrand factor, and then took that culture medium and applied that to valvular interstitial cells and monitored the production of calcific nodules. So they were stained with alizarin red S. And we controlled for histamine because that can also provoke calcification. And we found that really, regardless of the age of the VEX that these came from, that there were a lot of von Willebrand factor stimulated calcific nodules that formed. So this was exciting work, and we're building on that now. And one way we're building on it is try to analyze valvular endothelial cell behavior in not a valve leaflet, but on a platform where we can grow and control the cell behavior very precisely. And so our target was, how can we control the hemostatic behavior of valvular endothelial cells? And so we have been testing the valve endothelial cells that are grown on combinations of polyethylene glycol hydrogels of different stiffnesses. So a range of stiffnesses due to varying the molecular weight of the PEG, and then different ligands on the surface of the hydrogel. So a ligand derived from laminin, one that's reported to be more involved with thrombosis, and a laminin derived from fibronectin. And so we have been looking at a lot of different thrombotic and antithrombotic protein expression balance conditions, and both with gene expression and with immunostaining. And for example, here you can see that looking at the balance between von Willebrand factor and ADAMTS13, we get best balance where they're both expressed in these conditions where we have gels made from either 8 kilodalton or 20 kilodalton PEG and coated with the laminin ligand. And so the balance was quite different in all the other cases. And then if we follow up with a look at the way ADAMTS13 activity actually is able to cleave von Willebrand factor, we find that this particular group of the 8 kilodalton hydrogel coated with laminin-derived peptide best promoted in ADAMTS13 activity. So this is what we're going to be working with moving ahead in our further studies of VEC hemostasis. And so we also have been looking at the role of VEX in pathological angiogenesis, or just angiogenesis overall. So this is work that my student Alex is going to be presenting at a poster, so I'm not going to go over too much of this. But we've been looking at, first we looked at VEX alone, but more recently VEX and Vixen co-cultures. And we find that the network formation by VEX is a really dynamic process, and it occurs very distinctively from what you would expect from vascular cell co-cultures. So it's unique and interesting. And it's regulated by Rho kinase, protein kinase C, TGF-beta, and Tie2, not regulated by some other factors that we would have expected, but much more of this will be presented by Alex today on his poster, so I'm going to skip that. Okay, so since we've been working with hydrogels, we've started to also try to apply these towards VIC analyses. And so we've cultured VICs in 3D hydrogels that are either made from hyaluronan, like this is acrylated hyaluronan, or a biodegradable PEG by inserting a collagenase-sensitive peptide that we call PQ. And the cells are able to spread out. Some of them express smooth muscle alpha-actin. They express CD44, and especially for the ones grown in hyaluronan, they can express a lot of hyaluronan synthase, They make extracellular matrix, and we can measure how much collagen and elastin they are making. And my point from this slide is more that they make measurable amounts of collagen, that whether it's methacrylated hyaluronan, acrylated hyaluronan, or biodegradable PEG. And over time, they will make different amounts of elastin based on the specific type of scaffold that they're in. So there's a lot of potential for culturing the VIX in 3D. If we look at VIX in either 3D or 2D, we can see other ways that our scaffold formulation, our hydrogel scaffold formulation, will influence the VIC behavior. And so if we just grow VIX, valvular interstitial cells, in polyethylene glycol diacrylate that is not biodegradable, the cells will stay very round. But once you add in the degradable collagenase sensitive sequence so that the cells can degrade it, they start to spread out and migrate. And over time also in some cases express smooth muscle alpha actin. So the viability really increases as you increase to have a much more degradable hydrogel. And let's see, just moving on to some of the work that we've done in 2D, the valve, we can also culture the cells on top of a PEG hydrogel, and this is the amount of spreading based on the concentration of RGDS. So with increase in RGDS, we get, this is 0.3 millimolar, 1 millimolar, 5 millimolar, and cover slip. We actually, with the increase in RGDS concentration, we get increased spreading and slight reduction in circularity. And then also with the, I think this is 0.1 millimolar, 1 millimolar, and 5 millimolar, we get increased production of or expression of smooth muscle alpha actin. A lot of this isn't so new, but we're enjoying looking at seeing how we can show this work in our lab. And then what we've been moving towards is a co-culture model. So where we have valvular interstitial cells in the interior and valvular endothelial cells on the surface. And so this is, again, our laminin-coated peptide fragment attached to the surface. And this laminin coating was really important to prevent endothelial to mesenchyme transformation, which we actually try to avoid. I know it's really interesting, but we didn't want it right here. So we get it unless we coat the surface with laminin. And so then the VICs are in the interior, and we are able to keep the cells viable for a month of culture, and the different cell types will secrete and maintain their own extracellular matrix in the appropriate location. Like we have a lot of fibronectin production on the top, and then we have kind of this greenish yellow collagen fibers that form in the interior. So that's exciting, and we want to keep moving ahead with this co-culture model. Since I love biomechanics, we've been trying to move towards a more biomimetic structure for these hydrogels to have. And so one of the things that was impressed upon me when I was doing my dissertation by Karen Kunzelman was that it's important to replicate the bending mechanics of heart valves. And one of the reasons that they have unique bending characteristics is because they are softer in the middle. And so we've been working really hard towards getting a layered structure. And we published this last year, but we were able to get a softer glycosaminoglycan-rich interior of a hydrogel with distinct mechanical behavior from the two outer layers. And because of this layering structure, we are also able to also have different cell densities in the different layers, and we can also put different biological characteristics in the different layers. So, for example, a ligand from the peptide RGDS versus a ligand from AKVAV in the different layers. And then we are also working on patterning to add anisotropic behavior to these hydrogels. Also not necessarily a new technique, but we've been trying to now combine that with the layering. But what we're working on here is having reinforcements that are just due to a smaller molecular weight polyethylene glycol fragment being diffused in and then cross-linked via photo cross-linking. And we are able to get increased reinforcement and increased stiffness in the direction parallel to the fibers compared to perpendicular to the fibers in certain volume fractions. And we have been looking at different combinations of PEG of different molecular weights, as well as reinforcing methacrylated hyaluronan or acrylated hyaluronan. And really, heart valves aren't striped. Heart valves have a fiber splay of collagen fibers, but this is more of just a proof of concept. And we are able to achieve an anisotropic ratio that's comparable to what heart valves have, but these are still much stiffer than heart valves. So in order to try and make them stiffer, we've been moving towards doing some predictive modeling of hydrogel tensile mechanics in collaboration with a friend of mine who's a civil engineer who happens to be really good at modeling hydrogels. So we've applied several different models, whether it's a detailed pattern model where we discretely define these stripes, reinforcement patterns, or more homogenization models. And so I'm not going to get into this data so much as to just let you know that hopefully computational modeling can be used to improve engineered tissue development or scaffold development. And then the last thing that we've been trying to do with respect to scaffolds to try and improve their stiffness is to integrate an electrospin scaffold. And this is a technique that is fairly widely done in the tissue engineering community. And what we've done here is we created electrospun polycaprolactone, and then we modified the surface so that so electrospun PCl is actually quite hard to get the hydrogel prepolymer solution to soak into. So we modified the surface to make it more hydrophilic so that the polymer would just kind of wick in really nicely. And then we were able to grow valve cells on the surface to see if they would detect the characteristics of the underlying PCL scaffold. And so if we just grew the cells on polyethylene glycol diacrylate without any interior PCL, then the cells spread a little bit, but on the PCL plus on the PEG plus PCL, they actually spread quite a bit more, and their mean fiber direction for the stress fibers, this is just a phalloidin stain, I believe, really reflected the directionality of the underlying scaffold. So that was exciting. The cells can sense the material behavior of something that's actually quite a distance beneath them. Okay, just going to want to wrap up with what we've been doing for mitral valve mechanobiology. So some time ago, we have been comparing the cells that can be harvested from different regions of the mitral valve. Specifically, we looked at the cells from the center of the anterior leaflet, which normally experiences tension, from the posterior leaflet free edge, which normally experiences compression, and then the chordae. And just to focus on the two leaflet groups, we found that the anterior leaflet cells in the center really were very slow to adhere to tissue culture plastic. They made less collagen and they grew quite slowly in comparison to the posterior leaflet cells, which really grew quite fast, but weren't very complex in terms of expression of smooth muscle alpha actin or vimentin, or just overall internal complexity, which is a measuring of from side scattering on flow cytometry. And so then we looked at how these cells respond to changes in stiffness. So this is again using PEG diacrylate hydrogels of two different stiffnesses. And we did this by varying the weight fraction of the amount of PEG. So 5% versus 15% gives you about an order of magnitude change in stiffness of the PEG, both pretty soft. They're not as stiff as tissue culture plastic. And so then we had and then we also looked at the effective age. So we had pigs that were 6 weeks old, 6 month old, or 6 year old that we got the valve cells from. And we looked at the change in cell phenotype with respect to activation and expression of heat shock protein 47. And overall, we found that there was not one specific response of the valve cells to this change in stiffness. It really depended on whether the cells were from the center of the anterior leaflet or from the mitral leaflet. In some cases, we would get a reduction of stiffness, a reduction in a marker with increased stiffness. But that wasn't always the case. With the older valves, we found an increase in expression of heat shock protein 47. So, which led us to conclude that the mitral valve is very, very complex. And so now we've been looking at this more physiologically. So what we've been using is our Rice University flow loop system, which a student cleverly nicknamed Ruffles. And we use Ruffles a lot. So what we do is we take pig valves and sew them so that the mitral valve is attached to a gaskety structure up at the annulus. And then at the base, we have the ends of the tips of the papillary muscles so we can constrain the chordae. And we initially just had the chordae papillary muscles rather loosely constrained. Now we've gone to attaching them more towards this platform-based model so we can have precise control over where the chordae are. And then the mitral valve structure is put into a port that's in this mock left ventricle right here. And so we have these in an incubator, and we have culture medium flow down from a reservoir down into the mock atrium through the mitral valve. It gets ejected up through the aortic valve, which is just a mechanical valve, and then up to a compliance chamber, and then it circulates back to the medium reservoir. And so this is a nice organ culture system. We've been able to grow valves in here for up to 3 weeks under sterile conditions, although we more routinely do 1 or 2 weeks. And they are less expensive than animal models and more consistent than clinical studies, so we're happy with this. And we are able to achieve physiological pressures. Our flow rate is a little subphysiological, but we're still very happy with it. And we can fit 2 to 3 of them in a couple of incubators, so we are able to have a lot of repeats. And so what we've lately been working on is inducing mitral regurgitation. And so by having the tips of the chordae attached to a platform, we can move the chordae overall basally up towards the leaflet, apically away from the leaflet, in towards the center of the ventricle, or medially towards the outer edges of the ventricle. And so we use these we also can regulate the annular position by putting it onto a larger gasket. And so we've been creating 2 models, one of symmetric mitral valve prolapse created by moving the papillary muscles up towards the leaflet. And when we do that, we move from the blue curve here up towards the green curve, where we initially generate quite a bit of prolapse, and then that is regurgitation is sustained over a week. For the FMR model, we move the papillary muscles down and out, and the leaflet gets sutured onto a larger orifice. And so this color here, green, is the one I want you to look at. Here we have the increase in regurgitation over about a week here. So we get regurgitation with both of these models, as you do clinically, even though the chordae are moved in different directions. And so we can confirm with collaboration from my help with my friend Stephen Little that in the mitral valve prolapse model, we get an eccentrically directed jet, and the FMR model, we get a centrally directed jet. And so the effect on material properties is, it does also reflect what we see in clinical disease. So the mitral valve prolapse model, we have a reduction in stiffness of the leaflets and chordae. So the leaflets, stiffness and ultimate stress, and also reduction in stiffness of the leaflets. And this is accompanied by a reduction in material collagen that we show using a stain called Haravici, and an increase in GAGs and hydration that we measure biochemically. With respect to the FMR model, again, the mechanical properties match what we see in people, and so we have an increased stiffness and a reduction in ultimate strain, and we also have a reduction in hysteresis, and the chordae tendineae similarly show a reduction in extensibility and a reduction in ultimate strain. And overall, we see that there's a big increase in staining for MMP1, but then also an increase in staining for a lot of collagen synthesis markers and fibrillogenesis players. Prolyl-4 hydroxylase involved at the early stages of collagen synthesis, lyxyl oxidase involved in cross-linking, and decorin, which helps coordinate fibrillogenesis. All right, so just to summarize overall then, we've been using bioreactor and biomaterial platforms to understand the relationship between the external mechanics, the substrate mechanics, extracellular matrix, and the tissue and cell behavior. And we can develop distinct models of remodeling in mitral regurgitation just in 1 week, which I was skeptical that would happen, and I was proved wrong by my student. And we've also been looking at hydrogels because they are appealing as scaffolds for these biomaterial platforms. They have been less applied to heart valve tissue engineering compared to the wealth of work that's been done on D-cell valves or fiber scaffolds, but a number of us are starting to look very extensively at hydrogels. And hydrogels can provide the heterogeneity and extracellular matrix and structure that we see in the valve, but I think they require more work to be mechanically sufficient. So we'll see. I think the time will tell whether hydrogels are really a valid option for tissue engineering, but I think they're really good for biomaterial platforms. So, and another thing about hydrogels is that hydrogels can be biofunctionalized in order to make them direct cell behavior. And it remains to be seen if biofunctionalization will be compatible with modifications that need to be made for hydrogels to make them mechanically sufficient. So I'd like to thank you for your attention and also thank my graduate students who have done all of this work over the years and my funding. Thank you very much.
Great talk, Dr. Grande-Allen. Let me start the questions. So regarding the where you were patterning your hydrogels with either using photolithography or using the fibers, the PCL fibers, did you see any effect on the cells when you varied the width of these lines? Is that important?
I think it's going to be an interesting, that's an interesting question, and I would really like to follow up on that. We're sort of doing that work now because like there are limitations based on the way just light penetrates through them, so that influence how much patterning you're actually able to achieve that has a mechanical impact. And so this is why like multi-photon confocal based patterning is maybe a better way to go if you do want to have a lot of control. But yeah, I think that like the angle and thickness and length will be very interesting to follow up on.
Thank you. Any other?
Giovanni Ferrari, UPenn. Beautiful talk, a lot of interesting data, and a lot of questions. I'll choose one, which is related to the phenotypic stability of your cell when you grow them on polycaprolactam or hydrogel. We are doing a study like that using the human-derived valve interstitial cell, and one of the things that we want to investigate is the phenotypic stability, because you want them to be synthetic enough to produce the collagen to adhere, to grow, but at the same time, you don't want to push them towards the chondrosteogenic differentiation, because for future biomaterial and possible tissue engineering application, you want to retain the quiescent phenotype of the VSC. So I wonder if you have tested. I saw you show a 6 days time course. Have you checked the phenotypic stability? If they, I know these are poor sign, but did they show any kind of like osteogenic activation, and the synthesis that you are noticing is because the cells are adhering and growing and are trying to live on the surface, or because they are differentiating towards an activated form of the VSC?
Thank you. Most of what I can comment on is related to how the cells have behaved in a hydrogel scaffold, and we have actually had a rather hard time getting those to calcify. So we're trying to examine this in more detail, but classical osteogenic differentiation medium with dexamethasone and beta-glycerophosphate and ascorbic acid, we have had a hard time demonstrating collagen as indicated by a collagen stain, like using Xylenol, for example, to demonstrate it. And so now we're starting to look a little bit more into osteogenic gene expression. So I can just say that we've had a hard time getting them to calcify, which may be a good thing for tissue engineering. But for wanting to look at this as a model for looking at calcification mechanisms has been more challenging.
Thank you. Any other questions? If not, let's thank our speaker. Thank you.